Very good evening to all the NEET aspirants. This is your Prasanna Ma'am from NEET World. So yes, from our top 100 topics for NEET or concepts for NEET, today we are going to discuss about double membrane bound organelles from the cell, the unit of life. Okay. First you tell me what are the double membrane bound organelles that are involved in this chapter.
Chloroplast, mitochondria.
Yes, and chloroplast, mitochondria and no Kunal. Endoplasmic reticulum comes under single membrane bound. Yeah. So you have what? Mitochondria, chloroplast and nucleus. Fine. These are the double membrane bound organelles in a plant cell. If it is in an animal, cell, what are the double membrane bound organelles that you will see? Mitochondria and nucleus. Simple, right? Let us go in detail about each and every organelle. First, it is mitochondria. Mitochondria is cylindrical or sausage shape in structure. Okay, what is the shape of mitochondria? Cylindrical or sausage shape. Cylindrical or sausage shaped structure. Okay, this mitochondria is of course double membrane bound, that you know. First, let us look into what is the approximate diameter and length of the mitochondria. The length of mitochondria will be 1.0 to 4.1 micrometer in length. The diameter of mitochondria approximately is 0.2 to 1.0 micrometers in diameter. Okay. To understand this structure in detail, I will draw a cartoon structure where you will get to know it in much better way. You can note it down later, beta. Listen first. Okay. So let us assume this box as outer mitochondrial membrane. Remember all the time, outer mitochondrial membrane is totally permeable. Okay, and it doesn't have any invaginations or foldings. Yes, inner mitochondrial membrane, which is also called as cristae, has infoldings like this. Clear na beta? So you have outer mitochondrial membrane, this is outer mitochondrial membrane, and this is inner mitochondrial membrane. In between outer mitochondrial membrane and inner mitochondrial membrane, what do you have? Intermembrane space. Okay, what do you have, ma? Intermembrane space. Inside this inner mitochondrial membrane, what do you have? Mitochondrial matrix. Okay, this is what? Mitochondrial matrix. As I told you, outer mitochondrial membrane is completely permeable, beta. Yes, very good. This is freely permeable. And what will be the protein and lipid percentage in outer mitochondrial membrane and inner mitochondrial membrane? Okay. What is, like in outer mitochondrial membrane, what will be the lipid and protein concentration? Percentage. Okay. So protein percentage will be 60% and lipid percent is 40%. Clear? Next, in inner mitochondrial membrane, if you observe, what will be the protein and lipid percentage? Protein percentage will be 80% and lipid will be 20%. Clear? Yes. So here you can see outer mitochondrial membrane, which is completely permeable, inner mitochondrial membrane is selectively permeable, and this will be the protein and lipid ratio. Fine. Space in between, that is what? Intermembrane space. Inside you will have mitochondrial matrix. Now let us look into what will be the composition, what are present in the inner mitochondrial membrane and mitochondrial matrix. In the mitochondrial matrix, okay, in mitochondrial matrix, mitochondrial matrix, you will be having ribosomes. What type of ribosomes you will see in the mitochondrial matrix? Mitochondria and chloroplast, is it 70S or 80S type of ribosome?
70S.
Yes, it is 70S type of ribosomes. Okay, and
DNA material.
yes, very good, double stranded circular DNA is present. Double stranded circular DNA, 70S ribosomes, and ATP is synthesized. ATP is synthesized in mitochondrial matrix. Fine. ATP is synthesized in mitochondrial matrix. These are the three types that you observe in mitochondrial matrix. Clear, ma? So, this is the structure and this will be the composition. Clear, right? Next, in the inner mitochondrial membrane, whichever electron carriers and proton channels are present, those are present in inner mitochondrial membrane. Okay? In inner mitochondrial membrane, what are present?
Protein channels.
Very good. Or you can also say it as protein channels.
Electron.
Very good. Electron carriers. And else protein channels, electron carriers, and ATP synthase is present. ATP synthase. Fine. And there is a popular term where we call like mitochondria as powerhouse of the cell, chloroplast as kitchen of the cell. Why mitochondria is called as powerhouse of the cell?
ATP is synthesized.
Because ATP is being synthesized and it will store the ATP as well. So, you are clear with what are present in mitochondrial matrix, what is present in inner mitochondrial membrane. Clear, right? So, now here one more thing I would like to tell you is the inner mitochondrial membrane is having F0 F1 complex, right? That F0 F1 complex is also called as ATP synthase, okay, or oxysomes. Fine. What is that F0 F1 complex called as?
Oxygen.
Very good. Oxysomes. F0 F1 particles. Fine. Oxysomes, F0 F1 particles, and this is also called as ATP synthase channel. Fine. In this, the major point that you have to remember is F0 is embedded inside the inner mitochondrial membrane and F1 will be towards the mitochondrial matrix. Clear, right? That will be pumping protons from what? Intermembrane space to mitochondrial matrix. Clear, right? With respect to mitochondria. Now, if we have to look into the functions of mitochondria, okay? Functions of mitochondria. You tell me what is the primary function of mitochondria?
Synthesis of ATP.
How ATP is being synthesized? Through aerobic respiration. So, mitochondria are the main parts or the main segments of what? Aerobic respiration. So, this will conduct aerobic, very good, aerobic respiration. For aerobic respiration, why mitochondria is the key organelle? Let me give you a gist of it. Okay? This is completely cytoplasm. Inside cytoplasm, mitochondria is present. As we have discussed, this is what? Outer mitochondrial membrane, and this enfoldings. Very good. Yes, intermembrane space and mitochondrial matrix. In cytoplasm, we all know that glycolysis will take place. Glycolysis takes place in all the cells, be it prokaryotic cell or eukaryotic cell, both in aerobic and anaerobic respiration. Glycolysis happens in
Cytoplasm.
very good cytoplasm.
Glucose to pyruvate.
Very good. Glucose is converted to 2 molecules of pyruvic acid. This pyruvic acid will directly land into the mitochondrial matrix. Remember that. Inside mitochondrial matrix, 3 steps will happen. Okay. First, you will call it as link reaction. link reaction or oxidative decarboxylation. Okay, link reaction or oxidative decarboxylation, wherein two molecules of pyruvic acid will be converted to two molecules of acetyl CoA. This acetyl CoA will be starting that pathway called as TCA cycle. Okay, what is that called as, ma? TCA cycle or Krebs cycle. TCA cycle or Krebs cycle. Clear, right? So, first is glycolysis, then pyruvic acid will immediately land into the mitochondrial matrix. Yes, in here, no, not through acetyl CoA. This will directly come into the mitochondrial matrix. After that, oxidative decarboxylation takes place, then it gets converted to acetyl CoA and then Krebs cycle. Fine. After Krebs cycle, ETS will be activated, electron transport system. Fine. So, this is what happens in the mitochondria. This is how these are the sites of aerobic respiration. Second point is ATP synthesis, as you said. Okay, ATP synthesis. And third is oxidation of fatty acids. Okay, oxidation of fatty acids. Fine. These are the major functions of mitochondria. Okay, clear, right? So, you are clear with the structure and types, what are present over the mitochondrial matrix and what is present in the inner mitochondrial membrane and what are the functions of mitochondria, you are clear with it, right? So, now plastids. Okay. One of the plastid is chloroplast, but you know it better that plastids are of three types.
Chloroplast, chromoplast, leucoplast.
Very good. Plastids are found only in
Plants.
very
Plant
good.
cells.
Plastids are found only in plant cells, and you have three types of plastids. What are they?
Chromo, chloro, leuco.
Chloroplast, chromoplast and leucoplast.
Green color.
Very good. Chloroplast will be green in color because of its pigmentation, chlorophyll. But here, to understand the structure of the chloroplast, first you look into the structure of the chloroplast. It is again double membrane bound, but inside the chloroplast you have membranous sac-like structures. So, this membranous sac-like structure is called as what? Thylakoid. Stalk of sac-like structure is called as what? Grana. One grana is connected to other grana by stroma lamellae. Yes, one grana is connected to other grana by what? Stroma lamellae. These frets are called as stroma lamellae. Look into the labeling. This is the direct diagram of your NCERT. Okay, these frets are called as stroma lamellae. So, membranous sac-like structures, thylakoid. This thylakoid membrane is called as thylakoid lamellae or thylakoid membrane. Fine. This is called as thylakoid lamellae or thylakoid membrane, and inside this is called as thylakoid lumen. Fine. So, thylakoid lumen, thylakoid lamellae, that is how they make a membranous sac, thylakoid. Stalk is called as grana, one grana connected to the other by stroma lamellae. Inside this fluid filled space is called as stroma. Stroma are called as matrix of chloroplast. What is stroma?
Matrix of chloroplast.
Very good. Stroma is nothing but matrix of chloroplast. This stroma will be again having 70S ribosomes.
D circular DNA.
Very good. D not, double stranded, DS, double stranded circular DNA. So, I have a question for you. Double stranded circular DNA. Why does mitochondria and chloroplasts having same 70S ribosomes and double stranded circular DNA, same like that of prokaryotic cells?
They are derived organelles, or they came from outside the cell and
I'll explain you in detail. Firstly, they are prokaryotic cells only. As endosymbiotic theories explain, eukaryotic cells has engulfed this through endosymbiosis, and they have become part and parcel of eukaryotic cells. That you will call, that is explained under what theory? Endosymbiotic theory, important. Symbiotic theory. Initially they were prokaryotic cells, but they are engulfed by eukaryotic cells, that is called as endosymbiosis, which is clearly explained under endosymbiotic theory. And they have their own double stranded circular DNA, that is why they are called as what? Semi-autonomous cell organelles, semi-autonomous or self-duplicating organelles, self-duplicating organelles. Fine, semi-autonomous or self-duplicating cell organelles. The double-stranded circular DNA, which is present in the stroma of the chloroplast, and inside the stroma there are few more things which are present, like lipid droplets and starch granules. So yes, this double stranded circular DNA, what is the ultimate goal of DNA?
To transfer hereditary material to the offsprings.
Nothing
or
but
the functions.
No, the double stranded, be it double stranded circular or linear, whatever DNA, the ultimate goal of DNA is to synthesize proteins. That is explained in central dogma of molecular biology. So the ultimate goal of this also, this DNA is also what? To synthesize the protein. What protein is it synthesizing? World's abundant protein, that is rubisco, which is the world's abundant protein, okay, which is ribulose bisphosphate, carboxylase oxygenase, okay. So this is being synthesized from what? Double stranded circular DNA
of chloroplast.
Yes, clear with the structure of chloroplast. Now, where are these chloroplasts present?
In the plant cell.
What cell?
Plants.
What cell? In plant you have many cells, root cell, mesophyll cell.
Not meristem, permanent cells.
Chloroplast is present in the leaf cell, those are mesophyll cells. Mesophyll cells. And if you have observed, okay, some bacteria like blue-green algae, cyanobacteria will also conduct photosynthesis. Some unicellular organisms like eukaryotic protists like chrysophytes, diatoms, they will also conduct photosynthesis. Unicellular algae like Chlamydomonas, Spirogyra, they will also conduct photosynthesis. So is chloroplast number same in all those cells? No. Is the chlorophyll pigment same in all those cells? No. Okay. So chloroplast number varies from one cell to the other. For example, number and shape, okay, both number and shape varies.
Like stem, it has less chloroplast than
In stem you will not be having, only in young stems you will be having chloroplast. That number varies within the plant, within two different leaves of the same plant also. But let us assume between different species, like for example in Chlamydomonas, which is unicellular, right?
Yes.
Yeah, it has single chloroplast beta, okay, it will be having single chloroplast for one cell. Per one cell it will be having one chloroplast. Whereas in a plant cell, if you take advanced plant cell, chloroplast number can be up to 20 to 40 chloroplast per one cell. And as I said you, the shapes of the chloroplast also will be varying. For example, in Cara, it is discoid, okay. In Cara, you can observe discoid shape of chloroplast, okay, and cup shaped chloroplast you can see in Chlamydomonas. In Spirogyra, you can see spiral shaped chloroplast. Fine. This is completely about chloroplast. Chromoplast is with different pigments called as colored pigments, okay. colored pigments.
Carotenoids, xanthophylls.
Yes, colored pigments. It can be fat soluble pigments or water soluble pigments. You will be having two types of pigments, fat soluble and water soluble. Fat soluble pigments, as you said, those are carotenoids and xanthophylls. Carotenoids and xanthophylls. Carotenoids will give you like orange to yellow color, xanthophylls purely yellow color. Okay.
Red also carotenoids.
No, not red, orange only. Okay. Water soluble pigments, if you see, it can be phycobilins, phycoerythrins, those are water soluble pigments. Phycobilins and phycoerythrin. In rhodophycean members of plant kingdom, what do you see? Which pigment do you see? Phycoerythrin. That is why deep inside the oceans and seas also they can conduct the photosynthesis, because it has water soluble pigments. Next comes is what? Leucoplast.
Colorless starch.
Very good.
Granules.
Or not just the starch granules. Leucoplasts are colorless, as you said. Good. But then based on the storage product that which it is storing, we have three different types of leucoplast.
Alluroplast.
No, have patience. That is alluroplast, amyloplast and elioplast. Alluroplast will be storing up what, ma?
Starch.
No, protein.
Proteins.
Alluroplast will be storing up proteins. Amyloplast will be storing up what? Starch. Elioplast will be storing up oils and fats. Yes, good. Oils or lipids also you can say. Fats, oils and fats. Clear? This is all about the plastids. Understood? Okay. And there is one differentiation that I have to explain you. If you have to see F0, F1 particles, those in mitochondria, wherein F0 is towards the inner mitochondrial membrane, F1 is towards the mitochondrial matrix. The same thing if you see in thylakoid membrane inside that you will be having F0, and towards the stroma of the chloroplast, inside the stroma F1 will be present. This will be activated as a result of proton gradient, okay, where this is explained under what? Chemiosmotic hypothesis. This is also for the synthesis of ATP. Okay. The ATP synthesis which is taking place in chloroplast is said to be photophosphorylation. Okay, photophosphorylation. Whereas the ATP synthesis in mitochondria, you will call it as oxidative phosphorylation. Okay, in mitochondria, you will be calling it as oxidative phosphorylation. Fine. This is called as photophosphorylation because phosphate group is added to form ATP in the presence of sunlight. Okay. Whereas in mitochondria, it is through oxidation processes. So, it is called as oxidative phosphorylation. Clear, right? Kunal, everything is fine till here, right? Now, we will go to one more double membrane bound organelle, that is nucleus. Nucleus is double membrane bound, but there are 4 sites in the nucleus that you have to note. If I consider this as a nucleus, okay, of course this is double membrane bound, okay, the outer extensions of the nuclear envelope, this is nuclear envelope, these extensions will form endoplasmic reticulum, that is it. This nuclear envelope will be having pores called as nuclear pores. These pores are called as what? Nuclear pores. Inside the nucleus you will be having nucleoplasm and nucleolus. nucleolus. Inside nucleoplasm you will be having this genetic material which you will call it as chromatin. Till here this is fine now, the structural description of nucleus. Here inside the nucleus RNA synthesis will happen in eukaryotes. You tell me how many types of RNAs are there inside the eukaryotes. In eukaryotic cell how many types of RNAs can you see? Ribosomal RNA, messenger RNA, transfer RNA. 3 types, rRNA and tRNA. Where are they synthesized? Okay. No, inside nucleolus only rRNA is synthesized. Yes, only rRNA is synthesized. Inside the nucleoplasm both mRNA and tRNA are synthesized. Okay. tRNA is being synthesized by RNA polymerase 3. Okay. mRNA is being synthesized by RNA polymerase 2. rRNA is being synthesized from RNA polymerase 1. Clear, right? So, easy what they are being synthesized, and inside the nucleoplasm what do you see? Chromatin structure. The condensation of chromatin takes place in which phase of the cell cycle? Chromatin's prophase of the cell cycle. And here chromatin is what charged? The DNA of the chromatin is what charged? Yes, chromatin. DNA is what charged? Negatively charged. Do remember all the time. DNA is negatively charged because of the phosphate group in it. Okay. Negatively charged DNA is attached with positively charged histone proteins. Yes, histone proteins. This histone proteins are of 8 histone proteins octamer that we will learn in detail in mol bio. These histone proteins are what? Positively charged. These are having what amino acids? Lysine and arginine. These are the amino acids in the histone proteins. After complete condensation, you can see the clear shape of the chromosome, clear structure or morphology of the chromosome in which phase of the cell cycle? No, no. Structure of the chromosome, clear structure of the chromosome is seen in the metaphase. No. The structure of the chromosome is clearly visualized in the metaphase. Shapes of the chromosomes are clearly seen in the anaphase. Remember that. Okay. So, take down that statement. Shapes of the chromosome is clearly seen in the anaphase. Structure of the chromosomes is clearly seen in the metaphase. Now, we look into the chromosome structure, highly important. At the center of the chromosome, there is primary constriction. Primary constriction is also called as centromere. Fine, ma. Attaching to this centromere, there are 2 disc-shaped structures. Those disc-shaped structures are called as what? Kinetochores. Disc-shaped.
They help in attachment on spindle.
Very good. Yes. So, at the center you have primary constriction, also known as centromere. Attaching to that you have 2 disc-shaped structures called as kinetochores. And here in this chromosome, how many chromatids can you see?
4.
No, it is 2 chromatids.
Non-chromatids.
These are 2 chromatids. These are arms of the chromatids. Okay. So, you have 2 chromatids, each chromatid is having 2 arms. And there is one more constriction in the DNA, that is called as secondary constriction. This is made up of repetitive DNA sequences. That secondary constriction is called as satellite DNA. Fine. Are you clear with the primary constriction? Kinetochores, chromatids, arms of the chromatids, secondary constriction, that is satellite DNA. Satellite DNA is being used for DNA fingerprinting, okay, that you will learn in the DNA fingerprinting and human genome project. In clearly you will learn in mol bio, okay. So, this is about the nucleus structure, what is present over there, and the chromosome structure, fine. And as I said you, the clear shapes of the chromosomes are observed in the anaphase. If you have to look into 4 shapes of the chromosomes that are formed in the anaphase after centromere splitting. 2 arms of the chromatids, if they are equal, that forms out to be what shaped? V shaped. You will call it as metacentric chromosome, where 2 arms of the chromatids are equal and centromere is at the center. Centromere is close to one end, which forms one short arm and one long arm, that is called as submetacentric, fine, which is submetacentric, and this forms out to be what shape? Any guess? L shaped. Okay, this forms out to be L shaped. And next type, where you can see one very short arm and one very long arm, this forms out to be what shaped? J shaped. Okay, this forms out to be J shaped, that is acrocentric. Fine. Next is telocentric. One arm is very negligible and small, one is very long arm, fine. This is what? Telocentric, and this gives out to be I shaped, okay, which is telocentric. These are different shapes of the chromosomes. You should be clear with the names and with the position of the centromere and with the shape of each chromosome, clear? With this, we end up the session of double membrane bound organelles. In the next session, we are going to exercise cytoskeleton, cilia and flagella, and centriole centrosome structures as well. With that, we can finish off the most important topics of cell, the unit of life. Thank you.